Is my screen visible?
Yes, ma'am. Yes.
Okay. Yes. Yeah. So thanks again, Dr. Shruti, for the introduction. A very good evening to all of you. And it's nice to be here to discuss with you people on the clinical front, actively working with patients about what we do behind the closed doors of a lab towards the common mission of eradicating or managing anemia. So, as was introduced, I have a PhD in disease biology, more towards ion channels and channelopathies, which are responsible for a whole lot of diseases, ranging from cancer to arrhythmias. So these are proteins which are involved in a lot of physiological functions, but I'll not go into that right now because it's not in the agenda of today's discussion. So right now I'm the coordinator, head of the biosciences division at the AUKBC Research Center for Emerging Technologies in Anna University. And I also hold other positions there in the Center for Technology in Traditional Medicine, where we work towards looking at a more holistic way of treating various diseases. So I thought we'll begin today's discussion. I'll call it discussion because I'm not going to deliver a monologue and bore you out. I'll try to keep it as simple as possible. And I would urge that it be more interactive, so because we look up to you for a kind of guidance on where assistance from our side is needed so that we can work together on this. So once. Yeah, this is the customary slide we are supposed to begin with. So I thought, let me not miss that out. So as you all know, anemia is something which needs a very drastic attention by nutritionists and clinicians and every individual for that matter. So since you're all from a clinical background, as I gather, I don't think I need to tell you about the symptoms and the causes of anemia. So that will be a mere waste of time since you know much more about it than me. So let me directly. Yeah. So I'll directly go into what is known at the cellular level and at the tissue level and what we work on. So to begin with what is known, we know that intestinal iron absorption is a very highly regulated process, and human beings lose iron every day through urine and the enterocytes, which are dissociated from the gut and is passed out. And so they need to absorb that much iron every day. And it's also known that it's kind of iron is a very roguish element. So if left alone, it can cause toxicity to the cells, mainly because it generates reactive oxygen species, which cause havoc within the cells. So iron always have to be chaperone. So if you look at iron within the cell or the tissue, it's always found coupled with proteins. So if you take the serum, it's coupled with a protein called transferrin, whereas within cells, they are driven by other chaperone proteins and are stored as coupled with another type of protein called ferritin. So let's just go through what happens during absorption, because without this, I cannot explain to you what exactly we look at in the laboratory. So you all know that there are two types of iron which is ingested by any individual. You have the heme version and the non-heme version. So the heme version is normally absorbed by a type of, taken in, not absorbed, it's taken in relatively easily by a type of transporter which is present on the enterocytes in the gut. And the non-heme ferric iron is absorbed by the enterocytes only after it undergoes reduction. So for reducing it to the ferrous form, you need the action of a particular enzyme here called the duodenal cytochrome B, or it could be also ascorbic acid, yeah, which reduces it to the ferrous form. And only the ferrous form can go into the enterocyte, transported by a type of protein called the divalent metal transporter, DMT1. And this is very specific to the ferrous form. Now, once it enters here, whatever has to happen here happens, and either it's stored here and it's stored as coupled with a protein called as ferritin, as I told you earlier. So it stays there, or in case based on the levels of iron in the blood, it decides to be transported out. So when it has to go out, it passes out on the basolateral side, which is on the other side facing the circulation. And there you have a protein with the only known exporter of iron, which is called ferroportin. And ferroportin pushes out the, I would say, transports the ferrous iron out into the blood. And there it is again converted to ferric iron by action of various other types of oxidases, in this case, hepastine. And only then it can bind to plasma transferrin, which is again another of these chaperone proteins that I was talking about. And then it's transported to various organs in the body. So now this ferric form is a major form of iron for most cells. And where is it used? You need it for making the RBCs, where heme is needed, heme synthesis is needed. So how does it enter the cell? So you have this ferric form, which is bound to transferrin, binds to the transferrin receptor, and it is taken up into the cells. It is taken into the cells by a process called endocytosis, where the membrane pinches in, and you have the transport of this small vesicle, which contains the transferrin receptor bound to the ferric form of iron, which is in turn bound to the transferrin. So what happens inside the endosome? Endosome has various components which breaks this into bits, and the transferrin is released and the ferric iron comes out, and then it is exported into the cytosol. Cytosol by again another vesicular form of DMT1, which I told you in the earlier slide. So what happens is when there is a when there is no deficiency of iron in the body and the RBC production is going on normal, there is a type of hormone which is secreted by the liver called hepcidin, which mediates the internalization and degradation of ferroportin. So when there is no ferroportin, what happens is, if you recollect here, we have ferroportin, which is going to aid in the transport or pushing out from the enterocyte. So this is blocked or inhibited. So when that gets inhibited, there is no more iron coming out from the enterocytes into the bloodstream. So what happens to the iron which is taken in from the dietary supplementation or through the food? It is stored within the enterocyte along with ferritin. Now what happens is it waits there in case there is a depletion of iron in the circulation. You have this hepcidin whose action is decreased, and then it is transported as normal. But in case the person is good on iron, then this iron is not needed, and the enterocytes get slowed off. It detaches from there. It gets replenished, right? So then what happens is, after some time, the iron which is stored there is lost due to the deformation of the intestinal cells. So overall, if you look at the maintenance of iron homeostasis, you have the regulation of dietary iron by the enterocytes in the duodenum, the usage of this by erythroblasts. They are also stored by the hepatocytes, and you have these clinic macrophages which recycle these senesced RBCs, again providing a source of iron to the body. So we can say that the control of dietary iron absorption by enterocytes and release from the macrophages and from the storing hepatocytes contribute to the maintenance of iron homeostasis. So the major contributor, as I told you, is a liver-derived hormone called as hepcidin. And hepcidin secretion or transcription is upregulated, which means it is increased when you have increased iron levels, when there is an infection in the body, and when there is inflammation. So again, this infection is something you need to remember because I'll be touching upon it in a later on slide. So I just wanted to emphasize on it once again. Hepcidin binds to ferroportin, inducing its internalization and degradation, because of which there is a prevention of iron release from the enterocyte, which absorbs it from the food, or from the hepatocytes where it is stored, or from the macrophages where you have the recycled iron which is present. So this is what happens at the tissue level. So what is happening within a cell? How is iron sensed, and how does the cell respond to varying iron levels? So you have two very important moieties here. One is the IRP and the other is the IRE. IRP stands for iron regulatory protein and IRE stands for iron responsive elements. So what do these two do? They control the mRNA stability. So you know the central dogma of molecular biology, right? You have the DNA which gets converted to mRNA and the mRNA gets converted to proteins. So this particular IRP and IRE controls the stability of the mRNA and also the translation of the transcripts which code for proteins which are involved in iron uptake, export, transport, and storage. These include proteins like ferritin, transferrin, and all that. So you see there is a stringent control mechanism which happens even within the cell. So when a control of this sort is present, you see that the iron release from enterocytes, hepatocytes, and macrophages are prevented based on the situation. So this is just to summarize what is happening from a bigger perspective. We were talking at the molecular level, at the cellular level. So let's just run through it. So iron is absorbed as heme or non-heme iron. Heme primarily from animal sources, non-heme from plant sources. And the gastric digestion is very important because this helps convert the non-heme iron into a more absorbable form, which is the ferrous iron. And in the duodenum is where iron absorption primarily takes place. And in the duodenum, obviously, there is no acidic environment or milieu. So what happens is you have various enzymes like the cytochrome B reductase, which I told you about, or ascorbic acid, which helps in the reduction of the ferric ion to ferrous ion, which increases the absorption efficacy. So this is another reason why they say that you need to have iron along with vitamin C, which induces or enhances its absorption upon intake. So what happens after that? You have the ferrous ion, which is taken across the enterocyte membrane via the divalent metal transporter that I told you about. I'm not getting into details of that. So once inside these cells, these intestinal cells, the iron can be stored as ferritin, coupled to ferritin, or it is shunted out into the bloodstream, bound to the ferroportin. So in the bloodstream, again, it needs to be chaperoned, and you have a transport protein called as transferrin, which binds to the iron and takes it all over the place. So when you look at homeostasis, you can see that the body tightly regulates the iron levels, because either is a problem, deficiency in the case of anemia, or excess, which kind of rarely only happens, but we have anemia which is very prevalent. So hepcidin, I told you about that. Iron deficiencies, when there is low level of iron, the hepcidin production is decreased. So there is increased iron absorption from the gut, or there is increased release from the stores, which are the macrophages and the hepatocytes. When there is an iron overload, then hepcidin production is increased, which reduces iron absorption, and there is a facilitation of the storage of this iron in tissue. So as I told you, when the bone marrow makes RBCs, you have iron release from the source, because you need the RBCs when they are being made, you need iron for making the heme component of hemoglobin. And heme is a better way to absorb the iron, as all say. Then again, coming to macrophages, when these RBCs get old and senesced, they are phagocytosed by the macrophages. And this iron is again available for reusage within the body. Okay, so yes. So what is needed for this? A balanced diet is what is needed to ensure that there is no iron-related disorders due to a decrease of that. So this is known per se, but we have some unwarranted surprises which come in, which also affect people in the sense that there is impaired iron absorption when infected with organisms, parasites, or any other nematodes like hookworm, pinworm, or helicobacter. You see that what happens is they attack the body in a multi-pronged manner, which results in iron deficiency anemia. The patient or the person, individual, may be consuming ample quantity of iron, but still there is less iron actually prevalent in the body for executing the various activities. So what happens is these organisms may use up the iron for their own self. They reduce the intragastric content of vitamin C, which is needed for absorption, increase hepcidin synthesis. This was what I told you. When there is an infection in the body, the hepcidin levels increase. When the hepcidin level increase, there is decreased absorption of iron from the gut, and there is decreased release from the source. So what happens is there is a depletion of iron in the blood. And other than that, you also have these small gastric ulcers and micro erosions, which cause bleeding, which is again a form of loss of iron, which again results in iron deficiency, anemia in such cases. So this is the background of what is known. So what do we do in the research laboratory? I told you a whole range of molecules that are involved in the absorption and transport of iron, which help in the regulation of iron levels. So we do a lot of genetic study, in vitro studies where we overexpress, try to see what mutations may affect a particular individual, what makes it difficult for some people to absorb iron efficiently, even though the intake is good, and so on. And other than that, we also look at what are the factors which induce. So right now we stand in the era of micro RNAs and epigenetic modifications, which can influence these factors. But having said that, these are kind of more complicated ways for very serious cases. But if you take the percentage of people who have anemia, majority of it is because of decreased absorption from the gut. There is decreased absorption of iron in the gut. So I thought let's focus on the things that we can control, which is you cannot control what mutations are present in the body, but this is something which we as individuals, as consumers, as well as clinicians like you and scientists like us can have a look into and make the right choices. So one is the quantity. If you look at malnourished people, obviously there is decreased iron intake, which may lead to anemia. Another thing is the form of intake, the form of iron which is taken. Heme versus non-heme is the primary thing which comes to anybody's mind when you talk about this. And also, if you look at it, the way in which it is consumed. So I've had this a debate with many people about how ragi, is ragi a good source of iron or not? Because ragi is a rich source of iron as well as calcium. And calcium is an inhibitor of iron absorption. Same way you take, I mean, it's just a wild guess. You look at things like palak and paneer taken together. Okay, so is this good? Should it be adjusted? Can the addition of something like an acid, like ascorbic acid, or some kind of thing in these food preparations, can it remove this obstacle? Can it decouple this inhibition by calcium and other stuff? These are things which need to be explored. Another thing is the bioavailability, as I told you, is coupled to that, and associated factors like parasite infections, which hamper the absorption. So dietary factors, if you look at it, inhibitors, you have so many inhibitors of iron absorption, which include calcium, phytates, polyphenols, casein, which is present in most of the food that we consume. So it's about how a person's diet is planned. So that is very, very critically important, which is where both the nutritionists as well as the clinicians have a major role to play in identifying, advising the patients who come to them about how a particular form of food should be consumed to better the absorption of various nutrients. Let's not just talk about iron. So here again, though what you just need enough to maintain the loss of iron from the body, which is not significantly much. It's kind of easy to run on this, but still, if you look at the stats, you find that anemia prevalence is extremely high. In India, according to a recent survey, found that the current anemia prevalence ranges from 57.1 to 89.3%. It's not a number to be ignored. So it's shocking about how this is happening in spite of the awareness, in spite of supplementation which is given to children and to young adults, especially females in the reproductive age. But in spite of that, we find an increasing prevalence of anemia in both genders, in fact. So this prompted us with a group to think about. So we joined hand with a couple of nutritionists. So we wanted to do something using our expertise. So I told you that I'm an expert in membrane proteins and transport of ions, not just iron, but any ions, IONS ions, across the cellular membrane. So we got together and we kind of made a project, a proposal about how we could design a dietary reference index so that it can aid nutritionists and also help those who are making these nutritional policies and to introduce these good combinations in food fortification programs. So how is this bioavailability of iron studied? So yeah, before I go to that, so this project which got funded from the Department of Science and Technology to a tune of, they generously funded us up to 5 crores. We've got a team of us around 13 investigators. So we decided to look at anemia from various angles, including device development to developing tools to understand the bioavailability, a high throughput screen. So that's what is going on. We've just commenced the project actually a couple of months back. So I'll talk about that sometime later if anybody is interested. But how do we measure the bioavailability of iron? So when you make an advisory saying that, oh yeah, you need to mix this and that, you need to mix this food with that food to enhance it, how do you arrive at that? So the most common thing is the second point, which is the clinical trials. So you choose a cohort and you administer a particular combination food formulation to them, and then you measure the iron levels, measure the status, iron status, hemoglobin levels, and this and that, and then you arrive at a advisory saying that this combination seems to be better in absorption. So let's go ahead with that. So that is what has been used for centuries. And before going to that, what they do is they do the same thing in small animals, a murine model, like on rats or mice. You administer this formulation or iron mix with something or a nano formulation, you name it, whatever it is. It's done in vivo on small animal studies. So having said that, this is the primary way it is studied, but it's wrought with many difficulties because there's so much of variation and the cost is it's a whopping cost. So people started tuning to in vitro studies so that you can rank the food products which are developed by the nutritionist, and you can fine tune it in vitro rather than having to go for clinical trials and then come back to adjust one small feature saying that you need not have heated the ragi laddu so much at this temperature. So it kind of gets complicated at that point. So we finish all this in vitro and then you can move on to small animal studies and clinical trials. So what my team started doing is we started using this CACO2 cell model. So CACO2 is a intestinal carcinoma cell line, which has the ability to differentiate into intestinal enterocytes with polarity. And it is cultured on special inserts, which we have, and they are mounted on a layer. So you have a monolayer of the cells, which kind of mimics the gut lumen with the gut wall. Okay, so they are loaded with fluorescence dyes, and then we measure absorption of not just iron. You can, it's like a intestine on a chip, intestine in a petri dish. That's what we do here. So again, I just wanted to emphasize on this. How do you choose in vitro versus in vivo? So if you look at the duration of the assay, it just takes minutes to hours. How much time do I have, Dr. Shruti?
Ma'am, please go ahead. It is very interesting. Please take your time, 15 minutes
Okay.
more.
Okay. So what we do is, if you look at the duration of the assay, if you take an in vitro bioavailability assay, it takes minutes to hours. You have this preformed intestine sitting in the petri dish, you add, you measure the absorption. Whereas if you're going to do an in vivo assay, even including volunteers, it may take days to weeks to months for you to arrive at a small conclusion. And again, if you look at the variability is very high when you look at in vivo or clinical trials, because you tell the volunteer you have this at this time, they go have it, and then they want to have a chai. Yeah, immediately after taking an iron supplement, which you're specially given for the experiment, they just go and have chai, or they forget to take it at this particular time of the day. Some of them take it on a hungry, when they're hungry, and that may facilitate absorption, or there are various factors, variabilities. I need not tell you about it because you may be well aware of that. But these variabilities can be minimized if you're using any in vitro assay like what we do. And obviously, this involves in vivo or clinical trials involve animal and human testing, which means that you have a lot of paperwork. All the bioethics, biosafety, animal ethics committee, human ethics committee, every approval needs to be taken, which is like, it's kind of very time consuming. It's needed, it's very time consuming. It's a check which is very much needed. But that can be done at a later stage, because anyway you need to get to that, but not for all the products. You can shortlist, see which ones are good, and then move on to that. It saves on cost as well. So coming to the cost, there's high cost of experimentation if you're going for the in vivo assays or ex vivo assays or even clinical trials. Whereas this in vitro bioavailability assays that we do is very cost effective. It reduces the usage of animals, which is consistent with the 3 R's of animal ethics, which is kind of rampant these days. Yeah. So add-on is, which we recently discovered is, so we have a special chamber that we use. So instead of making the CACO2 intestinal sheets that we do by culturing a monolayer, we can also take biopsies, because I come to know that with my collaborators in various hospitals, gastroenterologists, they say that you have very complex kind of anemias in certain people. So they're not able to understand why iron absorption is not happening the normal way. They seem to be normal on every check, but even if iron is administered, they're not able to absorb. So is this something intrinsic, like as I was telling you, is it due to some kind of mutation which is present in the system, or what exactly is happening? So this, if we have biopsies, a small punch of the intestinal section, that area, we can actually put this on the device that I'm going to tell you about, and we can study. It's like kind of a personalized medicine, right? You look at the individual person's intestine, you can see whether absorption is happening the way it should, is it getting hampered? All this information is available very much in a very short period of time. So this is an add-on which can be done. It needs a lot of standardization and customization. It's not as easy as I'm making it a sound. But yeah, it's certainly very much doable. So yes, having said that, how do we begin the study? So we have to understand that we are looking at bioavailability in the duodenum. And in the duodenum, you don't get a bar of iron fortified food which directly goes there, or a raisin, or some dates which directly go in its live form. It's like Trishanku planning to reach heaven, right? In its entirety. That's not the way it works. So what happens is, first, the food products gets masticated in the mouth, and then it goes down into the stomach where it is digested, and then it goes to the small intestine. So we have to mimic this in the test tube, which is what we do. So we have something called as simulated salivary fluid, which has the enzyme, silin, and all that. We do all that, incubate it, mince it, homogenize it so that it resembles what happens in the mouth when you start chewing on a type of food. And then we put it through the simulated gastric environment, which has simulated gastric fluid, which again contains all the pepsin and various other components, the buffers, the salts, everything which simulates the gastric fluid. We let it sit there and incubate. Then we move it to the simulated intestinal fluid, where you have the action of trypsin, pancreatin, and various other enzymes, pancreatic amylase, with all the other electrolytes and things like that. So at the end of this entire experiment, which currently is taking us about a day, which we are working on to shorten the duration of, because ultimately our objective is to go for a high throughput screen. So we get a kind of like mix which resembles how the food looks like when it reaches the enterocyte where absorption happens. So yeah, so that's the beginning. So next is, what do you do next? So now we have this pre-digested food. So this is a kind of summary of what I told you earlier. So you have the gut lumen, which is in green, the enterocyte, which is spanning the gut wall, and then you have the circulation in the pink color here. So ferric ion gets reduced to ferrous ion, and it's transported into the enterocyte by the divalent metal transporter, and either it is stored coupled with ferritin in case the plasma levels of iron is high, or if the plasma levels of iron are low, it gets shunted out through ferroportin. This is what happens. So now you see this chamber which we have designed. So we have a mimic of that. We have a support here on which we grow. This is an optically transparent side, so that we can do the imaging under a microscope. This is a real image which I'm showing you of the Kakoto cells. You can see the nice finger-like projections, which is typical, and along with the polarity which is seen. So we grow that. In addition to it, what we are trying to currently do is we are growing it along with liver cells, because the liver secretes the hepcidin, which again influences the absorption of iron. So we co-culture this along with hepcidin. So on this side, so this top area represents the cis side, which is suggestive of the gut lumen, and the trans side is the circulation side. So we, on the circulation side or the trans side, we grow the HepG2 cells, or the liver cells, so that they can feel free and release hepcidin, and we can see what happens there. And the pre-digested food formulation, which is supposed to be in the gut lumen, we add it here. And then we wait for it to be we incubate it and wait for some time so that there is an uptake and absorption of iron, which we can study later on. And as I told you, here we have a clamp support so that we can put the intestinal biopsies there and then do whatever studies we want. So in this, there are various types of iron sensors depending on what exactly you want to do. So the project that we are working on, we chose to use a dye, fluorescent dye called calcine blue. So calcine blue has a sensitivity of being able to measure even up to 0.02 micromoles of iron, which is kind of very sensitive. So you can see that there is an absorption peak which is at 431 nm. So we measure that, and then we plot a standard graphs based on known concentrations of iron, and the fluorescence is measured at around 425, and a standard graph is plotted, and then we estimate the iron which is present in the unknown sample. So here, this is a depiction of what was done. This is a manuscript which is in preparation by my PhD scholar who's working on this project. So if you look at the first bar, it is supposed to represent the total amount of iron which is present in the media which is added. And the bar next to it denotes the iron which is absorbed. So this is added in the form of ferrous sulfate. Okay. So this was kind of like a testing which was done. And you can see that the absorption levels are pretty much good. It's more than 90% is getting absorbed in the model that we generated. It's an in vitro bioavailability model of iron, and it's made with the intestinal enterocyte-like cells. So then we started to look at, okay, let's see what happens if you add ascorbic acid, which is an enhancer. Okay, so, excuse me. Sorry about that. So here you can see that when we add ascorbic acid, there is a significant increase in the percentage of iron which is absorbed. So if you look at the graph on your right side of this, on the right side of the screen, you can see that the first bar denotes the total iron which is added onto the chamber. The iron absorbed is so much, but when it is treated with ascorbic acid along with iron, you can see that there's an enhancement in the amount of iron which is absorbed. So this is just the beginning that we are seeing. We are testing out various other things which due to confidentiality reasons I'm not allowed to present. So right now it is work under preparation. So basically what I wanted to say is that this is a kind of thing that we do. We look at mutations, we look at personalized medicine, biopsies, and see what are the various factors which impact iron absorption. At the same time, we are also looking at working strongly with nutritionists who develop these various food formulations, or you have these Nutri-bars which can enhance iron absorption. So we are working with them. And the solution to all this is not a single person or a group of people working as niches, but I think a team of clinicians, scientists, nutritionists, and most important, the food providers at home. They need to have an awareness of the right type of food combinations which needed to be done. And that is where Dr. Rajina's team, I would say, is doing an exceptional job in making people aware of what they are eating and how food should be consumed, so that you can maximize the benefits of eating healthy food and live a healthy life. So I think this collaboration is very much needed among all that. So as I just wanted to summarize, the right source for the food, the right combinations, and this is what does the trick in eradicating anemia. Yeah, so I think I'm done. Before I sign off, I just wanted to say that in case you want to discuss more, collaborate, you can reach out to me, or you can reach out to Dr. Rajina's team, and we can have a discussion. And if you want to look up some articles that I like to write as well, so some pop science articles you can look up. So I don't remember if I've written anything on anemia, but please feel free to look up my sites. Yeah. So that's about it. Yes, I'll be happy to take any questions.
Your expertise has inspired us to collaborate with more scientists to further illuminate the relationship between iron absorption and WFPB diets and vegetarianism as well. So Dr. Ashwini has asked one question here. How can the insight gained from understanding the iron absorption mechanism, as discussed in Dr. Kavitha's session, be practically applied by clinicians and nutritionists when advising dietary patterns for individuals, especially those following a WFPB diet and vegetarian lifestyle? So this question just popped up when you were showing that slide regarding that vitamin
Ascorbic acid.
C, vitamin C and iron. So I think, Doctor, we will be having more collaborative PPTs and session along with this. Would you like to answer this in your manner?
Yeah, I think as you said, I had kind of touched upon it. So major thing is what we are doing is we are developing in this project, we are trying to develop a high throughput screen to understand what enhances iron absorption in the gut. So now you have these nutritionists who make, like, we are working with a leading team of nutritionists, renowned people who are developing food formulations. So they may be making something like laddus, which have raisins, yeah, or some other thing, or maybe something based out of spinach and all that. So when they are making an advisory saying that you need to have it in this particular manner, so they will be tweaking because they are standardizing the protocol, or they are standardizing the formulation of the food that they are developing. So for each of it, they cannot go and do a clinical trial. They cannot do a small animal study. So that's when they come to us. So we do this study, tell them which enhances the absorption. Okay, you need to bake it at this temperature. Okay, don't heat it. Don't do this factor. Add so much concentration. You may add some more ascorbic acid, and they work around that, and they arrive at this formulation which enhances, which has maximum absorption of iron. Then this information is passed on to the clinician. I think that's what Dr. Ashwini asked. So the clinician is now aware, saying that they see, their job is to heal. They have to heal, and we are the back end support for them. So they are the front face who are going to represent all this extensive research which happens to the patient community, and they have to advise them and tell them, saying that this is what you need to have. You have it in this combination. And as I told you, Dr. Regina's team is doing a wonderful job. See, I know all the theory, but I have somebody with me who's pointing out every single meal of mine and telling me that, okay, this you need to cut down. See, all the theory I know, but you need somebody to watch over you. So that is where everyone, I think, needs help, whether we know it or not. So I think I answered your question, Dr. Ashwini. If not, please let me know.
Okay, any other question from students' end? Okay. So, ma'am,
I say that if there are no questions, it means either they understood everything or they didn't understand anything. So I hope it's a first.
No, we will be sharing this recording to our 1,000 students, medical students. So they must be having other questions after listening to this. So thank you so much, ma'am. Thank you so much. And I particularly enjoyed the discussion on inhibitors and the enhancer. And it was delivered in a simplified yet highly effective manner. Thank you so much, ma'am. Thank you so much.
Thank you, Dr. Shruti. Thank you, everybody. I'll take your leave because I've got another appointment now. Thank you so much.